Hello everyone, my name is Chao Ren, and this video is about our group's recent work on multi-resolution outlier pooling for sorghum cultivar classification. So I will get started from some details on the dataset we have worked with and the classification tasks that we are interested in. The first thing is the sorghum. We are focused on sorghum, a cereal crop that's widely used as a source of grain, agricultural feed, and biofuel. Here is some images of the sorghum. The next thing is the Terra Reef project. In 2016, the Advanced Research Project Agency Energy, or ARPAE, funding the installation of a state-of-the-art gantry-based crop monitoring system in Maricopa, Arizona. The transportation energy resource from renewable agriculture phenotyping reference platform, which is also called Terra Reef, aims to transform plant breeding by using remote sensing to quantify plant traits such as plant architecture, carbon update, tissue chemistry, water use, and other features to predict the yield potential and the stress resistance of crops growth in the over 1 acre field that is monitored. The gantry includes a large number of image sensors, including stereo RGB, thermal, short and long-wave hyperspectral cameras, and a laser 3D scanner, and collects thousands of measurements every day of the growing season. In the image on the right, we show the Maricopa field and the gantry, as well as some of the captured data projects. An image from the stereo RGB cameras, a depth plus surface normal visualization from the 3D scanner, and an image from the thermal camera. In this paper, we focus on the task of identifying which cultivar is seen in RGB images from June of 2017, which is midway through the growing season. In this season, the plants are quite large but not yield dodging or falling over. So the sorghum 100 dataset, which we will discuss in the paper, consists of over 48,000 RGB images from 100 different cultivars. Now I will state the classification problem. Accurately predict the cultivar shown in the image may seem like a straightforward task, but in fact that there is high inter-class similarities between different sorghum cultivars, making this a challenging fine-grained result classification task. It is a relevant task for growers. For example, error in the planting process such as not uncommon but can be a problem, especially in breeding and plant phenotype context. An automated high accuracy approach to determine a cultivar for a photographed plant can help to identify planting errors. In order to test whether proposed approach to this cultivar classification task generalizes well to unseen data, as opposed to overfitting the training set. We split the sorghum 100 dataset into a training dataset and a testing dataset. The Terra Reef field was designed so that the same cultivar was grown at multiple different locations in the field. We put one of these locations into the training set and one into the testing set, guaranteeing that the proposed approach are not overfitting to, like for example, states ground features. Now that we are familiar with the Sorghum 100 dataset and the cultivar classification task at hand, we are going to present our proposed approach to performing this task, multi-resolution crop outlier pooling. Our approach is based on using deep convolutional neural networks. In the common convolutional neural network architectures, one of the last steps is using a global pooling operation which converts spatially distributed features into a single number per channel. The output of the pooling operation is then commonly passed to one or more fully connected layers. The two most common species to performing this global pooling operation are global average pooling, which takes the average value of each feature channel as the output, and the global max pooling, which takes the max value of each channel as the output. Now we propose a new global pooling strategy, the outlier pooling. Briefly, in outlier pooling, we find the average of all activated locations for a particular feature channel that are above a threshold, where the threshold is a function of the mean and the standard deviation of the activation for that filter. In particular, we can see the feature output, the output at the feature channel J is determined by the average value of the locations in the preceding layer that's above the threshold. So the threshold is determined by the mean and standard deviation. So it's like mean plus the parameter multiplied the standard deviation. This is a parameter that we need to fine tune based on the data set. Intuitively, we can reason about why such a pooling strategy may be superior in the setting of the plant understanding. Consider an image of a plant with a couple of fast flowers and a filter that hypnotically activates the four flowers. In global average pooling, we overlay weight locations in the image that do not have flowers, downweighting the presence of the two flowers. In global max poolings, we only consider the one flower that most highly activated the filter. Outlier pooling, on the other hand, considers both flowers appropriately without underweighting them by overvaluing the activations on the non-flower locations. The next thing is the dynamic outlier pooling, which is an improved version of the outlier pooling. So we found that we could use this to improve the performance of the classification task by this. The dynamic outlier pooling approach, which will act more like average pooling early in the training process. So this will allow more locations to be considered in the average, and progressively has a harsher and harsher threshold, focusing more and more on outliers. So from the formula, we can see that in the early steps, the values below and above the threshold will have a similar weight, which are both close to 1. And with the progress goes, the weight of the value below the threshold will decrease to 0 gradually, and the weight of the values above the threshold will increase until to 2. So finally, this will gradually becomes to a typical outlier pooling. In our experiment, we found that the dynamic outlier pooling lead to very slightly higher accuracy on average, but noticeably faster convergence. So this plot is the conversion of the training accuracy of dynamic outlier pooling and outlier pooling by the epoch goals. And the dynamic outlier pooling converges faster, which means that it's accelerating over the typical outlier pooling. The final algorithmic contribution that we provide is the usage of two parallel branches prior to the fully connected layer and classification output. One branch is a low-resolution global scale branch, shown at the top of the network diagram on the right. In the branch, we resize the entire image, pass it through the network, and perform the global outlier pooling on the single crop. This branch allows the network to learn global scale features that are relevant for the crop as a whole. The second branch is a high-resolution local scale branch to which we feed multiple higher resolution crops. In this branch, we take multiple higher resolution crops from the original image and pass them through the network. The global outlier pooling operation then puts across all of the crops, considering outliers from whichever crops they came from. This branch is shown on the bottom of the network diagram and shows the network to learn more focused localized plant features, such as a particular texture on a leaf. We then concatenate the global outlier put features from both branches and pass that concatenated feature through the single fully connected layer, which is then softmaxed to retrieve the predicted sorghum cultivar. The next is our implemental details. In terms of specific implementation detail, we used random fitting models pre-trained on ImageNet for each of the branches. The global scale's low-resolution image is generated by resizing the original image to be 512 pixels on the shortest side and then taking a random 512 by 512 crop during the training and a 512 by 512 center crop during the testing. The local scale high-resolution crops are generated by resizing the shortest side to be 1026 pixels and then taking 4 random 512 by 512 crops during the training and the 4 non-overlapping 512 by 512 crops from a 1024 by 1024 center crops during the testing. We normalize by channel means and standard deviation and perform random horizontal and vertical flips and data augmentation during training. To analyze the performance of our approach, we performed an application study comparing across different cropping strategies as well as different global pooling strategies. The standard approach would be use either average or max pooling with a single resized whole image crop. This approach performs significantly worse than using either multiple crops with no low resolution branch or both high and low resolution crops across all pooling strategies. For this particular dataset, average pooling outperforms max pooling, while our proposed dynamic outlier pooling significantly outperforms either across all cropping settings, which means that our multi-resolution dynamic outlier pooling method is more suitable on this particular dataset. Finally, in conclusion, we presented the Sorghum 100 cultivar classification dataset, which includes RGB imagery from 100 bioenergy lights of sorghum that are grown in the tariff belt. We additionally presented the approach that achieves over 78% classification accuracy on the dataset using a multi-resolution crop architecture and a dynamic outlier pooling method. Additionally, we welcome anyone to try their approach on this problem using Sorghum 100 dataset and the challenge that's available on the Kaggle page. We additionally hosting a contest on Sorghum biomass prediction, which is being hosted by the CVPPA workshop at this year's ICCV, and welcome any competitors. That's all, and thank you for your time watching this.